This is a bacteriology tutorial on bacterial genetics presented by the SchrDbb reference Lippincott Illustrated Review of Microbiology by Cynthia Cornilesson, Bruce Fisher, and Richard Harvey. So, you're going to visualize now bacterial genetics. So, you need to know that generally bacteria is made up of a nucleoid, which is made up of chromosome containing DNA. Now, I want to speak on this general diagram on the bacterial genome. So you need to know that generally the bacterial genome is made up of chromosome and it's made up of the plasmid. The chromosome is a circular double-stranded DNA, and it is double-stranded DNA and it is circular, unlike in a eukaryotic cell, which is not circular and which is entangled inside the protein called histones to produce the nucleosomes, and then which is going to later on produce the chromosome with the sister chromatid. So it's going to be made up of about 3,000 genes. Is it clear? So you have 3,000 about 3,000 kilobases. So we have the next is going to be single copy per cell. There's a single copy of chromosome per cell, and it is highly folded in cells. Now the plasmid now is a circular double-stranded DNA, just like the chromosome. But in this case, it's not made up of up to 3,000, but it's made up of 5 to 100 genes, and we have 1 to 20 copies per cell, and yet it's only a single copy per cell. Is it clear? Now, the next diagram here is a bacteriophage replication. So what is a bacteriophage? A bacteriophage is just a virus which is capable of infecting and controlling the metabolism of a bacteria. Is it clear? So how does bacteriophages act? So we start by having attachment of the phage to the cell based on its receptor. That's a bacteriophage which attaches to the cell wall of the bacterium based on its receptor. Is it clear? At about 0 minutes, that's what happened. The second now, we have the injection of the phage DNA into the cell. So when the DNA is going to enter into the cell. So that is number 1 minute. The later on, we have synthesis. So it's going to take the control of the bacteria by its DNA. So you are going to have now the synthesis of the phage protein. Is it clear? And when you have synthesis of phage protein, it will occur at the level of number 10 minutes. Then you have now assembly of the new phage particle. Is going to be, you have complete phage particle occurs at this level here, and you have lysis so this is going to occur by the level of the number 25 minute, and at the 30th minute you have lysis of the cell and the release of the progeny of the fish. So as you can visualize here. Is it clear? So that is how we have the cell cycle based on the bacteriophage. So now in this diagram now we have visual detection of bacteriophage in a plaque method. So how can you detect a bacteriophage on the plaque? So if you have a mixture of 10 raised to the power 8 uninfected bacterial cell and a single phage infected cell in a melted agar, you have solidification on top of the agar layer immobilizes all the cell. Is it clear? So you have that when you put them on agar, you're going to solubilize all the cell. Then later on, 12 hours later, you have phage replication in the infected cell. The cell releases the progeny of the intact adjacent cell. This cell releases and the cycle repeats. Is it clear? So you are going to have growth of uninfected bacteria create an opaque except in the location of the original infected cell. So what they are just saying is that you are capable of doing you are capable of detecting a bacteriophage just by this method here. So if you want to detect a bacteriophage, you can just place a plaque that is going to attach the bacteria on a particular slip here on a culture media. So you see that in this culture media, where there is going to be the phage bacteria, because the phage bacteria is going to be at this position, there is not going to be the growth of bacteria at that level where the phage bacteria is going to be present. Is it clear? That's why at that level on this plate here, on this agar plate, which is very good, which is a very good culture measure. There is no growth of bacteria at this level because it is phage virus. Is it clear? So that is can be capable of in use in the detection of phages. Now, when what involved in a cell-to-cell transfer of conjugative plasmid, how does it occur? So, in order for you to do a cell-to-cell transfer of a conjugative plasmid, you need to know have two main types of cell. You have an F positive and an F negative cell that is going to make contact together. Is it clear? The F positive cell is what is going to have the plasmid, while the F negative cell do not have the plasmid. Is it clear? So they are going to contact together by the use of their pili. When they have contact together by the use of pili, you have one strand of the plasmid is going to be nicked. Is it clear? So that is the strand is elongate and the 3' end. So as you can visualize, you need to know that you have two main ends here of the plasmid. The plasmid is made up of DNA, and the DNA is made up of two main ends. You have the 3' and then have the 5' end, and it is double-stranded. Is it clear? As you have seen up here, you said the plasmid is circular double-stranded DNA, as you can see. So, so you have that plasmid down here. So this plasmid down here is going to be double-stranded. Now we have two main parts. The 3' end is going to remain in the F positive cell, while the 5' end is going to move up to the F negative cell. Is it clear? Now we have the single strand is going to cut after one complete circle is transferred at the same time. The complementary plasmid strand is going to be synthesized in the F negative cell. What they are just saying is that when you need to know that normally there is production of the DNA replication occur in the 3' to the 5' direction. So you see that it's going to elongate in the 3' to the 5' direction. Though the 5' direction is going to enter into the F negative cell. So as it elongates, you're going to have plasmid. So you're going to have the formation of the DNA replication is going to elongate too in this direction, such that here you also have a 3' to 5' direction, and here you have a 3' to 5' direction, and at the end you have the end or are ligated, restoring the double-stranded circular configuration, and each cell, both of them become F positive cells. So this diagram is going to show you that you have certain fish that can package bacterial gene and transfer them to other bacteria. Is it clear? So this is a method of transmission of different gene by phage virus and not by the help of the plasmid. So a phage virus can infect one bacteria. When you infect one bacteria, this is the bacterial chromosome, it can penetrate into the bacterial chromosome. The phage replication and fragmentation of bacterial DNA. Is it clear? So when it causes fragmentation, you have now lysis. So when there is lysis, you need to know that there are normal non-transducing phage that are going to be produced, and when there is also lysis of that bacteria, you can see that there is sometimes the DNA of certain bacteria can enter certain phage virus, and that DNA that gene is going to be called the A+ gene. Is it clear? And that A+ gene now can enter other bacteria to give them the potential to cause infections. Is it clear? So this A+ gene can enter other bacteria. Example, so this you have if you have UV induction, normal excision of the prophage, and you have rare abnormal excision of the prophage. You see, when the bacteria is going to infect, certain are going to have, and they are going to release normal non-transducing phage, and then others, when they are going to be released, they are going to release phages that are going to have the S+ gene, as you can visualize them. So the next diagram here is just to show a replicative transposome that can move from place to place. Is it clear? So you need to know that at stage 1, transposome at the original site can jump to the other site, leaving behind a copy of itself. and at stage 2 you have transposome can jump into and out of a plasmid. So the transposomes are just part of the chromosome that can move from one part to another. Is it clear? So it's left inverted repeat here. So you have a transposome. There are two genes that two genes control the movement of transposomes, which are the transposase gene and the resolvase. Is it clear? And this is the antibiotic resistance. So they can jump from one part to another, and when they jump from one part to another, they can lead to the formation of plasmids with those transposomes. So the next diagram here shows 3 common mechanisms of antibiotic resistance. The first mechanism here is the antibiotic sensitive cell. So this is the antibiotic sensitive cell. Antibiotic is going to target the cell here. So the antibiotic is going to be targeting this particular part of the cell here. So you are going to have the first mechanism is going to be decreased permeability. So the antibiotic cannot enter again the cell. That's the first thing. Second mechanism is alteration of the target. Normally, let's say that the target of that antibiotic is a circle. If the change of the circle become a rectangle, the antibiotic is not going to target again that particular part. So you have alteration of the target, and the next one you have inactivation of the antibiotic inside the cell. So those are the different mechanisms. Now we have bacterial gene can be controlled negatively by repressor or positively by activators. So what does it mean? Mean that so you have different so you have negative control and you have positive control of bacterial gene. Is it clear? So you need to know that the bacterial gene is made up of different parts. You have the repressor gene, you have the promoter gene, you have the operator gene, and you have this part here is a lac-operon gene, which is going to be the main one that is going to be transcribed. Is it correct? Now we need to know this is the repressor. The repressor is what is going to bind. So we are binding of repressor to the operator to prevent the expression of the lac-operon gene. So as I've said, the lac-operon gene is the part that is going to be transcribed to give the metabolism of the bacteria. And now the repressor, the promoter, and the operator gene are the one that are involved with the control of this bacterial metabolism based on the genetic aspect. So the repressor gene actually is going to produce a certain enzyme that is going to repress at the level of the operator gene. So when it binds at the level of the operator gene, now it's going to inhibit the activation of the operon gene. Is it clear? And that operon gene is what is going to cause now metabolism. Now the next one, now we have the inducer. Also we have, in case where you have a certain inducer like lactose. Is it clear? Lactose is an inducer of bacterial growth. Is it clear? So when lactose is present, the lactose can bind to this normal enzyme that is produced by the repressor gene. So when it binds to the enzyme produced by the repressor gene, the repressor enzyme lactose complex is capable of now preventing the operator gene from being inactivated. So the operon gene is going to be transcribed to produce the lac mRNA. So this is when the repressor gene is going to be inactivated. So those molecules, example lactose, are called inducers. Now in positive control, this is negative control mechanism. So with repressor, and when you use inducers, it inhibits the negative control mechanism. Now in case of positive control, you see have the operon gene here, and you have the operator gene. So we have the operon because of the repressor is active. So the repressor is active, is going to inhibit this. Is it clear? So operon also here is off because the first case is because the repressor is active. Now operon here is off because the repressor is active. The cap is irrelevant. So we also have a cap here, CMP complex. Now the operon is on because the repressor is inactive and the cap is active. Is it clear? So this one is inactive. So the cap, the CMP complex, serve as an activator of the lac-operon. So I just want to tell you another aspect on the fact that the promoter gene can also activate the operon gene. So the promoter gene can produce the CMP, the cap CMP complex, which is capable of activating the transcription here. Is it clear? So if both if the cap now is inactive, you don't have any activation of the operon gene. Is it clear? So what can cause the activation of the operon gene is the presence of the promoter gene. The promoter gene activates the operon gene with the presence of the cap, while the repressor gene inactivates by the producing a certain enzyme that is going to inhibit the operon gene, operator gene. Is it clear? So all these are the factors which are involved in the positive control and the negative control mechanism. Negative control mechanism by the repressor gene and positive control mechanism by the operator gene, by the promoter gene. So we now pass to the next chapter of the day, that's Staphylococci.